Okay, this is where I want to pick up right here, just kind of picking up where we left off last time, comparing how a toilet flushes to all those principles of neural transmission that we went over. So, little note to the sub here, yes, we took every student in this class, we went into the bathroom, we flushed a toilet, it was a class field trip, it was awesome. So, you guys worked in pairs, you worked at your tables, kind of going through these, and how it relates back to what we were talking about with a neuron and it firing. So just to point out a couple of things here, the threshold. Just like when you're pushing down the handle of a toilet, it's got to reach a certain point before it actually flushes. Same thing with a neuron. It has to reach a certain threshold before it will flush, a certain stimulation. So, resting potential, same thing there. Once the toilet fills back up with water, it's ready to flush again. It's just waiting for somebody to push the handle. Just like when you're talking about a neuron, it has potential once the positive ions get sent back outside that selectively permeable wall of the axon, and it's just waiting for the cell body once again to tell it to fire so it can go on to the next. These are some big terms that you need to know. I put these in your notes handout. Just to go along with the firing of a neuron, action potential is the actual firing of that neuron, the impulse traveling down the axon. Ions, remember, outside the membrane of that axon wall, there are positively charged ions on the outside and negatively charged ions on the inside. So when that selectively permeable wall kind of opens up and allows those positive ions in, it creates a chemical reaction that causes the impulse to travel down the axon. Resting potential and selectively permeable, just went over those. Depolarize, depolarization is the term that we refer to when those walls open up and the positive ions are let in. Refractory period, remember that means that once that ion has activated, then it can't activate again for a certain amount of time. Each neuron's different, but there is a time after it's been stimulated and it's fired down and had that impulse that a time has to pass before it can do it again. Excitatory versus inhibitory. Once again, referring to how every neuron is different. Some are excitatory and the threshold is lower for it to fire and shoot an impulse down. Some are inhibitory, which it's going to have a higher threshold where a higher threshold has to be reached before it will fire. Basically, they're lazy, okay? And the all or none response. All right, so let me explain what we got here. Hold on, everybody got their dendrite branches there? So down here, we have the dendrites. They only receive information. That's all they do. And if the information they receive reaches a certain point, let's say our threshold is 2 ping pong balls, okay, then this neuron will fire. If it doesn't reach 2, it will not fire. And remember, it's only going to fire one direction. It's going to fire that direction. And then down there, we basically have Riley kind of holding the terminal branches here. They're going to send it on to the next dendrites. Okay? So let's see what happens here. Hold on. We have a very inhibitory, lazy neuron here. It does not want to fire. I got on purpose, by the way. It did not want to fire. No balls were caught. So that was an inhibitory neuron. It did not fire because we didn't get the mandatory 2 ping pong balls for it to fire. So please pick up the ping pong balls. Let's try this again. Throw them in here. Okay, so once again, all right, get your dendrites there.
I love it.
So once again, we need 2. We need 2 ping pong balls to be caught, put into the cell body before this thing will fire. Okay? Let's see if we can do a little better job here this time. Okay. Hold it with one hand. You got one hand open. Get the good catching hand open. All right, here we go. Come on. Now, excitatory neuron. Everybody's waiting for something. Here we go. All right, throw them in. How many we got? We got 1, 2, 3, 4. It's going to fire. Pull it tight. Watch your fingers.
Did you see that?
Move your fingers out of the way. There you go. We didn't get one. All right, have a seat. All right, now moving on to something different here. We're going to talk about how individual neurons communicate. Last time I kind of told you that all these neurons are connected to other neurons, and this just makes this huge web of neural pathways in your body. Technically they are not actually connected to one another. We thought they were for years and years, and then kind of some recent discoveries have shown us that they're not actually connected. There's actually a small little gap that we call the synapse, or the synaptic gap, that is the junction between the tip of a terminal branch that's sending messages onto the dendrites of the next neuron. So we call that the synaptic gap, and inside this gap, basically these chemical messengers are sent across, which we're going to call neurotransmitters. And there are several different types of neurotransmitters, each one having a kind of different effect on the neuron. But when these neurotransmitters are released, they get sent across that synaptic gap, and when they reach the dendrites, those receiving branches of another neuron, then those chemical messengers just kind of carry on that impulse. And then you guys know how it takes the impulse down the dendrites into the cell body, and it decides whether it's going to make that individual neuron fire. Another term that goes into this is called reuptake, and this is a neurotransmitter's reabsorption by the sending neuron. So, just kind of taking a look at what this looks like. There's a good image in your book if you look, I think it's on page 58, where it shows you, these chemical messengers, these neurotransmitters, which in this case would be the little green balls that are traveling across the synaptic gap, attaching to, the receiving terminals of the next neuron. And that is going to be what carries on that transmission from one neuron to the next. You take a look at this little chart. Once again, this chart's in your book if you want to look at it. But it shows reuptake a little bit better. If you look at kind of the last thing on this little chart on the far right, it talks about the sending neuron sending across those chemical messengers, the neurotransmitters, and then once they have done their job, they've communicated to the next neuron, there's a process called reuptake, where all of those excess neurotransmitter molecules are reabsorbed back into the sending neurons branches. So that gives you a little bit better idea what that looks like. So I told you there's a bunch of different types of neurotransmitters, and this is something that you're going to have to know. Not all of them, but the ones that really relate to psychology. Here are just a few. And if you want to follow along in your book, there's a really good table on page 57 that shows you what each one of these do. Acetylcholine is one that we understand a lot about. We kind of know what acetylcholine, or ACH, does, and the main thing that it does, the main thing that we see that it does, is it is basically a motor neuron. That it's between every single motor neuron and a skeletal muscle. And when acetylcholine is released, the muscle contracts. It's basically the chemical messenger that makes your muscle contract. So when a motor neuron sends out the message to move a muscle, the acetylcholine is the neurotransmitter that does that. Dopamine and serotonin are really big in psychology, talking about things like emotion and hunger and mood and sleep and arousal and all that. Dopamine, you probably remember from Psych 1, because we talked about dopamine being linked with schizophrenia. If you have an excess amount of dopamine, it can be linked to schizophrenia. Serotonin is a big one that we talked about as a mood-enhancing neurotransmitter. So an undersupply of serotonin could be linked to depression, and is in a lot of cases. And then we get into things like norepinephrine and GABA and glutamate and endorphins, and all these are going to sound really familiar to you, those of you that have taken anatomy. But we'll make our way through these. Like I said, this was part of your reading last night, and there's a really good chart on page 57 if you want to look and see what each one of these do. I'll kind of make reference to a few of them as we go through here. Understanding what neurotransmitters do. And a lot of times, drugs that we take, whether it be an antipsychotic medication or even something that gets rid of a headache for us, like an Excedrin or a Tylenol, anything like that, they basically are targeting neurotransmitters. That's what drugs do. They alter and they affect neurotransmitters. So let's talk about the two ways that they can affect neurotransmitters. The two ways that they can affect, and the drugs that we take, are either agonists or antagonists. And agonists, basically what happens is agonists are so similar to an actual neurotransmitter that it mimics the effects of that neurotransmitter. So, a great example is endorphins here. And endorphins is a naturally produced drug that, basically happens in your body, when you're exercising or when you're, in a good mood, your endorphins are pumping and it makes you feel good. It kind of dulls pain. Well, morphine, which is a man-made drug, mimics endorphins. It's so similar to the neurotransmitter that is the endorphin that it mimics the effects. So, when people are in the hospital and they're in pain after surgery, a lot of times doctors will give them morphine because it mimics the effect of endorphins. Now, long-time abuse of something like morphine that mimics the effect of a neurotransmitter can lead to that neurotransmitter actually not being produced as much. So people who get addicted to these things like morphine and painkillers actually do need them because they've taken them so much that now their body naturally tries to balance itself and it says, okay, we're producing too much of this, we need to cut back, when actually it's a man-made drug that's producing those effects. So when it cuts back on its natural production of endorphins, the people need to keep taking morphine to dull the pain away because your body has a natural natural state of balancing itself out. Antagonists, okay? These are only similar enough to bind to the receptor sites and block those neurotransmitters from attaching. So they're not similar enough to the neurotransmitter to actually mimic its effects. They are only similar enough to block the neurotransmitters, receptor sites, so that the actual neurotransmitter can't get in there and attach to those sites. So, great example here would be when you're in the hospital. When a doctor gives you anesthesia, basically what it's doing is it's blocking acetylcholine. It is paralyzing you. So where you can't move your muscles and you kind of feel like you're knocked out or don't have control. Those numbing agents that doctors sometimes give you, even at the dentist office, are blocking acetylcholine so you can't move your muscles. Another example the book gives is Curare, which is this same type of idea where this ancient tribe used Curare to put on the tips of their needle or the tips of their arrowheads when they were shooting arrows. And basically what it did was when it would hit an animal, that Curare would get into their system and once again block the ACH, paralyzing the animal's muscles, and then it would make it easier for the people to track down those animals and kill them since they couldn't move. So those are the difference between agonists and antagonists. A better look at this, and once again, this is a bigger picture in your notes if you want to look at it. Let me check what page this one's on. This one's actually on page 58. When you have an antagonist and an agonist, what you have is, just take a look at this. The very top one would be the actual neurotransmitter molecule. Fits perfectly into that site. You move down, the agonist, very similar. That's why it mimics the effects of that neurotransmitter, but not exactly the same. And then the very bottom one you see, only 1 out of the 3 is similar. That's the only reason it's hitting that site. But it stays on there and blocks any neurotransmitters from getting in there. So once again, when you're talking about certain neurotransmitters being linked to psychological diseases, we said an excess amount of dopamine could be linked to schizophrenia. So what do you think an antipsychotic medication does? Exactly. It's an antagonist. It gets in there and it blocks the receptor sites from receiving that dopamine, therefore stopping some of those hallucinations that people with schizophrenia have. All right, I wanted to use this quick little 3D medical animation to kind of show you what all these neural pathways look like, and taking a look at how all these things are interconnected. And then you go up close, this would be the synaptic gap right here, and as you kind of see, it shows some of those neurotransmitters being sent across the synaptic gap, which is kind of cool.